In this video, we are going to be looking at a series of scenarios and determining how we can calculate dilutions and concentrations of culture that we are going to be working with in our microbiology lab. In this first situation, we are going to be finding out how many cells are in a given volume. So the scenario is that we are given a culture and you are told that there are 2 times 10 to the 9th cells per milliliter. If you transfer 100 microliters of that culture to a plate, how many cells total would you be transferring? So in order to calculate this, I'm going to be using our equation, concentration multiplied times our volume is equal to the total number of cells. Now, how I know to use that equation is because if we see our question is asking us how many cells total would we be transferring, and this is the equation in which we can utilize to calculate total cells. And so we are given our concentration of our culture and our volume that we are going to be using. And so we are able to just plug these numbers into our equation. So our concentration, 2 times 10 to the 9th cells per milliliter, or per 1 milliliter. Multiply that times our volume, which is 100 microliters. We can kind of say that's over an imaginary 1. And that will give us the total number of cells that we have. And so if we were to do this math, we would have 2 times 10, and because we have 2 zeros here, we can think about that as adding 2 tens to our exponent. And so we would have 2 times 10 to the 11th. But what you'll notice is that our units are inconsistent. So if we were to transfer our units, this would actually be cells per microliter cells times microliters over mils. And we know that we're just trying to calculate our number of cells. So we can cancel out these additional units that we do not need by multiplying this number, 2 times 10 to the 11th, and that'll be cells per microliter over 1 milliliter, times essentially 1. So our conversion is, we know 1,000 microliters is going to be equal to 1 milliliter. And we need to cancel out our mils and our microliters. And in order to do that, we would then put 1,000 microliters in our denominator, so that would cancel out with the numerator. And we could add 1 milliliter to our numerator to cancel out the mils in our denominator. And thus, what we would end up with would be 2 times 10 to the 8th. Now, how I got that number is because instead of multiplying times 10, which would increase our exponent to 11, by dividing by a series of tens, in this case, we could also think about 1,000 as 10 times 10 times 10. So that would be 3 tens. We would subtract from our exponent by 3, which would give us 8. So our answer for this question would be 2 times 10 to the 8th cells. Now, a way to skip past all of this dimensional analysis would be to immediately convert this 100 microliters that we were given to the units of interest. And so, another way to write 100 microliters is to say that that is 0.1 milliliters. And that's using this conversion that we have down here. So, another way for us to calculate this, and I'll write this off to the side, would be 2 times 10 to the 9th cells. That's over a mil. Multiply that times 0.1 milliliters, and that's just over an imaginary 1. And essentially, we would get 2 times 10, and now, because we are multiplying times a decimal, another way of thinking about this math is that we would be dividing by 10. So, because if we're dividing by a series of tens, we are going to be subtracting from our exponent. We then end up with 2 times 10 to the 8th cells. And our units cancel out on our first try. And so what you'll see is that there was a long way to get the correct answer, but if we convert our units early on, we can also get the same answer a little bit quicker. So now, moving forward, we can calculate concentration if we have the number of cells in a given number of liquid. So kind of doing the reverse of what we just did. Again, we're going to be using our equation, which is concentration times our volume is equal to our number of cells. And another way to write this equation, in order to isolate for our concentration being equal on one side, would be to say that concentration is equal to our number of cells divided by our volume. Because we are given the number of cells plus the volume of broth or culture that we used, we can simply plug these numbers into our equation, which would be 200 cells divided by 100 microliters of broth. But knowing that we'll want this value in milliliters, I'm going to go ahead and skip a step and convert my units right here, because 0.1 milliliters is the same thing as saying 100 microliters. I can then easily do this math. So you can either plug this value into your calculator, or think that if you're dividing by a decimal, that's the same thing as saying we're multiplying times 10. So our number would be 2,000 cells per milliliter. Now, in order to write this number in scientific notation, as we saw up above, what we do is we take this number and find a number between 1 and 10. So if we move our decimal point, we can see if we have one digit before that decimal point, this would give us 2.0, and then we multiply that times 10. But because we moved our decimal point 3 places, that's like saying 2 times 10 times 10 times 10. So another way to write that in proper scientific notation would be 2.0 times 10 to the 3, because we have 3 tens. And always make sure to write our units, which is going to be now cells per milliliter. In our next scenario, I'm going to be showing you 2 different ways in order to calculate initial concentration. So for starters, we're going to work through this problem the long way using our equation CIVI equals CFVF. So the information we're given is that we take 200 microliters of a culture and we add it to 800 microliters of saline. The concentration of the new culture, or the diluted culture, is 1 times 10 to the 4th cells per milliliter. So we need to find out what the concentration of that 200 microliters of culture was before we added it to the 800 microliters of saline. So in order to plug in our values into this equation, what's helpful for me is to identify what variables that were given in our problem. So the first question is asking us what the concentration of our starting culture. So we already know that at the end of this, we are going to be trying to solve for our CI. We also know that the volume of this initial culture started out with 200 microliters. If we look at our problem, we are given the concentration of our new culture or our final culture, and so we know that CF is going to be equal to 1 times 10 to the 4th, and that is in cells per mil. And then finally, we know what our final volume was, and we have to do a little bit of math, but because we know we started out with 200 microliters and we added that to 800 microliters, our final volume will be 1,000 microliters, but we also know that that's the same thing as saying 1 milliliter. So now we can plug these numbers into our equation to be able to solve for our initial concentration. So if we just kind of rearrange this equation, that would give us CI being equal to our 1 times 10 to the 4th cells per mil. Multiply that times our 1 mil, and all of that will be divided by our volume initial, which would be 200 microliters, or to convert this number would be 0.2 mils. And so what we can determine is that we can cancel out some of our units. So we can cancel out this mil with this mil because they're both in the numerator. And then now we're left with 1 times 10 to the 4th cells divided by 0.2 mils. And we know that dividing by a decimal is the same thing as multiplying, and in this case, it would be the same thing as multiplying by 5. So the answer that we would come up with would be 5 times 10 to the 4th cells per mil. Now, how I figured that out was because if we think about 0.2 in a fraction, another way to say that would be 1/5. And if we are going to be multiplying by a whole number, another way of thinking about that is by inverting our numerator and denominator. And what you'll have noticed, if we go up above, the same thing is true if we had been dividing by 0.1 mLs. When we're talking about multiplying times 10, the reason why is because 0.1 in a fraction form is the same thing as 1/10. So we multiply times that denominator. Thus, multiplying times 5 gives us our answer, the same thing as dividing by 0.2. So this is the first method in order to solve for our initial concentration, but I would argue that there were a lot of steps that we needed to do in order to get to our answer. And there's actually a quicker way to solve for our concentrations, and we've kind of talked about this a little bit when we were calculating concentration from colonies. So the other method in order to calculate concentration is by using what we call our dilution factor. And simply put, we know how much we are diluting a particular sample, and so we can calculate backwards how much our initial concentration would have been, knowing how much we diluted it by. So if we know that we started out with 200 microliters, and in total we ended up with 1,000 microliters, we also, if we remove some of these units, cancel some of these zeros, it would be the same thing as saying 2/10 or 1/5. And so we can look at this number and recognize that we must have done a 1-to-5 dilution or a 5-fold dilution. And so another way to think about this is if we know that this concentration here must be 5 times more diluted than our starting culture, a quick way to solve this is to calculate what 5 times the concentration of our final culture would be. So in this particular instance, multiplying our concentration by 5 would give us a concentration 5 times as concentrated as our culture. And that number would end up being 5 times 10 to the 4th cells per mil. And what you'll notice is, again, we solved and got the same value, but the second way was a little bit quicker. So now we are going to move on to talk about how we would do dilutions. In this particular scenario, we are given 2 concentrations. So using that same culture above that had a concentration of 2 times 10 to the 9th cells per milliliter, how would you dilute that culture to get 1 milliliter of a culture with a concentration of 4 times 10 to the 8th cells per milliliter? So, because we are given two concentrations and we are going to be solving for a volume, immediately we know that we can use our equation CI VI is equal to CF VF. And again, to go through, we need to identify which variables we have and which ones we're going to be solving for. So we know that our final concentration is going to be 4 times 10 to the 8th. So that would be our CF. We also know that we want to end up with 1 mil of that culture, so we also know our final volume. Now we're given one other number, and we know that our starting concentration was going to be 2 times 10 to the 9th. Thus, we have our CI value as well. And so, what that's left us with is figuring out what volume of our culture that we will initially need to take from our starting culture to dilute that into 1 milliliter to get a culture that has a concentration of 4 times 10 to the 8th. And I was bad and I did not write my units, so I'm going to go ahead and fill those in now. Okay, so we are going to plug in our numbers. And so, because we are solving for VI, our equation will be CF, so that's 4 times 10 to the 8th cells over, again, that imaginary 1 milliliter times 1 milliliter, because that's our final volume, and we can say that's over an imaginary 1. Now all of this will be divided by our starting concentration, which is 2 times 10 to the 9th cells, and that also is going to be over 1 milliliter. So, in order to simplify this, the first thing that we can do is canceling out our milliliters shown here. And then what we are going to do is begin to simplify our equation. So here we will now have 4 times 10 to the 8th cells divided by 2 times 10 to the 9th cells, and that's all going to be over 1 mil. And so, because this is just over 1 milliliter, we can kind of forget about this 1. And what that's going to give us is now essentially 4 times 10 to the 8th divided by 2 times 10 to the 9th. And so, how we do this math is we can break up our first half of our equations and our second half. So 4 divided by 2 is just going to be 2. And then how we divide our 10 to the exponents is by subtracting them. So another way to think about this would be 8 minus our 9 in our denominator, which would give us a -1. So we can write this number as 2 times 10 to the -1. And now we got to fill in our units. And so what this will end up looking like is cells, and that's going to be over cells per milliliter. Well, in honesty, we can cancel out our cells, but this gives us kind of a weird situation where we have our whole equation over basically a fraction. Just like we were talking about above, where if we divide by a fraction, it's the same thing as multiplying, we actually can bump this milliliters up to the numerator, and it would be the same thing as saying 2 times 10 to the -1 mils. And this would be a hard number in order to pipette or think about, and so what might be more helpful is to first write this into standard notation. Because we have an exponent to the -1, that would be the same thing as moving our decimal point one place to the left. And so another way to write this number would be to say 0.2 mils. And we also know that 0.2 mils is the same thing as saying 200 microliters. And so now we know that our answer for how much culture we would need to take to dilute into 1 mil is going to be 200 microliters. Now finally, we're given a scenario where we will need to complete a multi-step dilution. In this scenario, we're given a bacterial culture that has a concentration of 8 times 10 to the 7th cells per milliliter, and our teacher asks us to dilute this culture to 4 times 10 to the 4th cells per milliliter. We are given 4 tubes, some sterile saline, and we need to now describe how we would approach this task of diluting. So, the first thing I would do is write out our concentrations. So we know that we're starting with 8 times 10 to the 7th. And we need to get to 4 times 10 to the 4th. So the first thing that I will do is compare the exponents. So we have our starting concentration being 10 to the 7th, and then we have our final concentration being 10 to the 4th. Now, what I'm going to do is take my exponents and subtract them to determine the difference. So that is going to be a factor of 3. And so we can think of that as being 10 times 10 times 10. And because we are given 4 tubes, we would be able to dilute our culture 3 times by a factor of 10. But we also need to remember the number that comes before our 10 to the exponent, and we have a difference of 8 and 4. So what I'm going to do here is figure out how much I would need to change my culture in order to get the actual concentration that we want. Because if we were to just do 3 10-fold dilutions, the number that we would end up getting to would just be 8 times 10 to the 4th cells per mil. Because we're factoring in our exponent going from 7 to 4, but 8 times 10 to the 4 is more concentrated than 4 times 10 to the 4th. And so how I would do this would be to then take our two numbers, so we can just kind of ignore our 10 to the 4th right now, and I would divide them. That would give us an answer of 2. And so what this number will tell me is that I still need to dilute this culture that we calculated here by a factor of 2, or by 50%, in order to get the culture that our teacher had asked us for. And so in order to do that, I would then have to do a 2-fold dilution. So the question is asking us to describe how we would approach our task. And so right now, we've figured out that we'll do a series of 3 10-fold dilutions, followed by a 2-fold dilution. We need to figure out what this math will be and what we are actually going to put into our tubes. So what I would recommend is drawing out our culture tubes, because I'm a very visual person, and so this would be very helpful. And then I would draw out our concentrations, because we know that that is going to be starting out as 10 to the 7th, and our final concentration will end up being 4 times 10 to the 4th. So I'm now going to plug in the fact that we're going to do a 1 to 10 dilution here, another 1 to 10 dilution here, a third 1 to 10 dilution here, and then finally a 2-fold dilution, or a 1 to 2 dilution. And so now, when we're reading this problem, we're not given any volumes. And so we can sort of make things easy on ourselves, and we can just say that each tube will have 1 milliliter of liquid in it. To do a 1 to 10 dilution, that would mean that we would take 100 microliters of culture, or 0.1 mLs of culture, and we would then have 900 microliters of saline. That would equal 1 ml. We would then transfer 100 microliters of that into another tube that had 900 microliters of saline. We would then transfer another 100 microliters from our second tube to our third tube, and this tube would also have 900 microliters of saline. But now we're stuck with a new problem, which is, how do we do a 1 to 2 dilution? Well, essentially that would mean that we're taking a tube, and this new tube, when we're all said and done, will have half of it being our liquid from our previous tube, and half of it would be new sterile saline. And so, if we keep our total volume at 1 mil, essentially what that would be is 500 microliters of saline, and then transferring 500 microliters from tube 3 into our final tube. And so, another way to sort of figure all of these numbers out is by thinking about our total volume and multiplying it times our dilution factor. So, if we know we're going to end up with 1 mil, we could then multiply that times 1/10. That would give us 0.1 mLs, which is the same thing as our 100 microliters of culture. And in the case of our 1-to-2 dilution, or our twofold dilution, we could do the same thing. And that would give us 0.5 mLs. And so, by showing our work and showing this math, we have answered the question of how we would go through our diluted tubes to get our new concentration of 4 times 10 to the 4th. Doing 3 10-fold dilutions, followed by 1 2-fold dilution.